So without further ado, I will pass you on to Disha, who will be speaking to you about quality control and differential gene expression analysis. Over to you, Disha.
Hi, I'm Disha. I'm a junior in high school, and I was a part of the June Bioinformatics session this year. I'd like to just start off by thanking my leads, Erin, Isha, and Eves, for an incredible experience and for the help getting started in bioinformatics. In my section of the presentation, I'll be focusing on the quality control and differential gene expression analysis steps of the pipeline, which we learned through recreating the results of a scientific paper. So, one of the first trainings we had was about how to read and understand a scientific paper. As an example, in what order should you try and read a paper? Skim the abstract, read the introduction, results, and conclusion, and then go back and read the abstract again. I found that this method of reading the paper was really helpful for comprehending the challenging material found in the paper. We also learned about the GEO database, which is a repository of curated gene expression data, and NCBI PubMed, which has the latest bioinformatics papers. You have free access to both of these NCBI sites, so they're a great resource for anyone interested in bioinformatics. Moving on to the project itself, the aim of this paper, which we used, was to find prognostic markers for colorectal cancer. Prognostic markers, by the way, are biological characteristics that are indicative of the likely health outcomes of a patient, such as disease recurrence. I will explain the required steps of the pipeline and the R-Bio conductor packages involved in the next few slides. But first, let's talk a little bit about the process of generating gene expression data. So how do we get this data? We use microarray sequencing, where an experiment with a single chip or array can provide information on thousands of genes simultaneously. Each dot that you see in the image is referred to as a probe, and it represents a specific genetic sequence. In microarray expression analysis, the sample has genes from both the normal as well as the diseased tissues, which are distinguished using dyes. The brightness intensity of a probe after hybridization denotes the gene expression levels. And that light intensity is converted to a numerical value, and that is the data which we will be examining. Now, in quality control, we have two main goals. One is to check the reliability of the microarray sequencing, and two is to identify and remove outlier samples, which may be caused by biases in the experiment. The RLE and news plots, which are shown on the right, are from the AFVPLM package for normalization. Let's focus on the RLE plot for right now. So most of our data will have similar expression levels, so if we can isolate probe sets that appear to have significantly different expression levels, we can eliminate possible outliers. If you look at the RLE plot, you will clearly see that the last probe set is an outlier compared to the other samples. So there are many quality control packages as listed under the main tools section, and it's a good idea to use multiple packages in order to get a high level of quality control, because some plots may miss certain outliers. Personally, I like the AFVQC report package the best, as it provides several helpful plots within just one function. Now that we've cleaned up the data, we are ready to start DGE analysis. First off, what is an expression level and why is it important? Expression levels are the rate at which a gene is transcribed. So if a gene is involved in a disease, you may see high expression levels called an upregulated gene or low expression levels called a downregulated gene. If the gene isn't involved in the disease, you shouldn't see any variation in the expression levels between a healthy and diseased sample. We did DGE in 3 steps: annotation, gene filtration, and lemma analysis. Annotation is simply linking the biological information to the numerical data. Gene filtration is removing a large part of the data so that only the most significantly differentially expressed genes remain, and in lemma we fit a linear model to the expression data for each gene. So there's a lot of intriguing math involved in DGE analysis. We don't need to understand all of it right away, as it does get rather complicated, but I would just like to cover two key concepts, p-value and fold change. Here's a demonstration of these concepts using a volcano plot. A volcano plot is a type of scatter plot that shows statistical significance, or p-value, versus the magnitude of change, or fold change. It enables quick visual identification of genes with large fold changes that are also statistically significant. These are likely the most biologically significant genes. As an example, the highlighted gene HOMER1 is down-regulated and statistically significant, so it is a gene of interest in our research. So returning to the paper we started with, shown on the right, some of this may begin to look a little bit more familiar to you and make a little bit more sense. As you can see at the top of image A, the researchers started with 3 of our data sets from GEO. Then in image B, they identified the upregulated and downregulated mRNAs. Images C and D are the visual representations of what was found, with image C being a type of volcano plot like we discussed earlier, and image D being a heat map, which is a plot that shows correlation between different genes. On the left, I attached a snippet of our code that we used to perform the steps needed to recreate these graphs. And Dan will begin to tell you more about the next step, which is functional analysis. So overall, I faced significant challenges in this internship. For one, because I came in with less understanding of biology than used in the paper, I initially struggled with understanding the bigger picture. And also because I was new to programming, I had a hard time with the different object types and figuring out how to manipulate them. And I think my major achievement in the session was overcoming both of these challenges. Also, working with other people of different experience levels who also enjoy bioinformatics was a really great experience, and I would like to thank the leads and STEMaway once again. I hope this session was informative for future students, and if you have any questions, please feel free to leave them in the Q&A chat, and we will answer them after Dan's part of the presentation.